For our final design project, our group decided to make an mRNA vaccine for influenza. The influenza vaccine is one that needs to be modified and manufactured every year due to the changing genetic nature of the influenza virus. While the current demand is met, this proves to be a costly and time-consuming process. Currently, the influenza virus is made by either live attenuated viruses or whole inactivated viruses, and thus have varying recovery rates. Also, due to the various strains of influenza that exist, the efficiency rate remains low. In 2018 to 2019 flu season, the overall efficiency was 29%, with the lowest effectiveness of the vaccine being 10% in 2004. mRNA vaccines are more attractive as they are cheaper to manufacture and can also be produced more rapidly. Furthermore, mRNA vaccines are considered safer as the virus does not need to be handled during the manufacturing process. Thus, this reduces the risk of infection. Additionally, there is no risk of insertional mutagenesis as the mRNA strand does not enter the nucleus. Rapid production of mRNA vaccines provide the potential for a faster response to outbreaks such as the coronavirus, and in our intended case, a quicker time for strain identification to the vaccine for the flu season. The influenza virus could be made utilizing a different platform, not only would reduce costs, but allow for a timely response to pandemic, but may also create the potential for a vaccine cocktail that combines multiple strains of influenza, and thus providing a more robust vaccine against the seasonal flu. Conservations of the influenza strains such as matrix 2, nucleocapsid, matrix 1, or stem of HA proteins can be selected. Specifically, the stem region is relatively well conserved among many HA proteins, as indicated by the low mutation rates of surface proteins. It is thought that HA stem will induce the formation of protective neutralizing and non-neutralizing antibodies, which can then activate cytotoxicity, phagocytosis, and T-cell immunity. In fact, current research limits the effectiveness of HA stem-derived vaccines to within the same HA group. Thus, only partially universal vaccines can be created for different subtypes of the influenza virus. However, by utilizing a cocktail approach, it may be possible to combine different vaccines into a single dose to be administered to a patient for a truly universal vaccine. This proposal aims to combine the idea of a universal candidate for the influenza using an mRNA vaccine cocktail to create a rapidly producible vaccine to combat many of the challenges. Furthermore, the cocktail could be also induced adjuvants to increase T-cell immune response if the mRNA candidate cannot induce a strong enough immune response with one dose. At the bare minimum, however, mRNA sequences for the HA stem for all HA groups of the influenza will be targeted and combined into a single cocktail vaccine. Here's the overview of our upstream processes. The first step in the upstream processing is the creation of a template DNA plasmid. The template DNA plasmid will encode for glycoproteins, hemagglutinin or HA stem, and neuraminidase NA for the most circulated flu strains of that season. HA is the most commonly targeted glycoprotein in most flu shots currently, and neuraminidase has been shown to improve immune response. The template DNA is amplified in E. coli and subsequently purified and linearized. This is then followed by cell-free in vitro transcription and 5' capping. To produce mRNA, a genetic sequence from a DNA plasmid containing the sequence coding for the antigen is mixed in a reactor with nucleotides and enzymes. The enzymatic reaction produces the mRNA using a DNA plasmid template, and the mRNA is encapsulated in a lipid nanoparticle for delivery. The use of self-amplifying mRNA will increase the potency of the vaccine over time. A self-amplifying mRNA encodes the antigen in addition to the viral replication machinery that will allow for intracellular amplification of the RNA sequence once it is introduced into the host cell.
Because we do not expect the bottleneck of production to be in the upstream processes, we will ultimately scale up to a 5-liter batch bioreactor. Process development studies will first be done in shake flasks and then translated to bench-scale bioreactors. Shake flask cultivation will be done in 500 ml with 100 ml defined medium. For the production of plasmid DNA, E. coli host strains of K12 and B type are typically used, but we will use the DH5 alpha for higher yield. Next, batch fermentation will be tested using a 1 liter working volume where cells will be grown at 37 degrees Celsius. A complex medium will be used with an initial glucose concentration of 20 grams per liter. Previous results from other studies demonstrated that pDNA tilters in the range of 150 to 250 milligrams per liter and specific productivity with DH5 alpha at bioreactor scales was approximately 8.02 milligrams pDNA per grams DCW. A similar study utilizing high cells density fed batch fermentation in 3.7-liter benchtop bioreactors with 2 liters cell culture volume. If we use this ratio for scale up, we will have a 5-liter working volume and a total of 9.25 liters. So a total of 10 liters total bioreactor volume will be used. It is expected that there may be decreases in productivity at higher volumes due to the change in surface area to volume ratios.
This is the overview of downstream purification processes that are currently being utilized for mRNA vaccines. Our proposal differs, however, because we include a continuous production and purification process. We propose to cut down on processing time by creating a continuous downstream process, which can retain and recycle high-value raw material, which could possibly provide a substantial cost reduction. It increases utilization of production line and increases efficiency. Unfortunately, at this time, the upstream process portion needs to remain a batch reactor rather than a continuous one, as it is directly connected to the downstream process, and thus a flow rate cannot be induced within this bioreactor. This is something to consider for future work. We propose to have a continuous system where the production occurs in a temperature-stable bioreactor, placed above the purification columns, and have the production flow into the column after production occurs, thus removing the need to remove and transport product. This process would have to be tested and validated as there is limited information currently available on mRNA vaccine production. This could also potentially decrease points of contamination and reduce costs as well. Here, the transcription reaction utilizes DNA-dependent RNA polymerases, which elongates the RNA transcript as it moves along the DNA template. The template DNA strand contains a positively charged particle, and by inducing a positive charge on the columns, the mRNA transcript can be separated out from the template DNA as the mRNA transcript will not contain the positive charge after transcription of the template. A cap is added to the 5' end of the mRNA, which protects the mRNA from intracellular degradation. The method of its in vitro transcription reaction will be used for the synthesis of mRNA, which should yield a 99.5% pure RNA after purification. In order to minimize any inhibition of enzymes by the presence of others, only enzymes that are used in the specific timeframe will be activated. Since capped DNA is removed with heating, we can control the activation and deactivation of enzymes by taking advantages of changes in temperature. Specific ligands can be attached to the poly-A tail enzymes that can be removed with increasing temperature. In this way, the capped DNA will be released simultaneously with the activation of a poly-A tail enzymes, allowing for a continuous production schedule without having to add substrate ingredients during production. The purification of the mRNA sequences after synthesis is critical as it directly impacts the potency of the vaccine. Any contaminants such as enzymes, residual template DNA, double-stranded or DS transcripts, and etc. must be removed and specifically the removal of dsRNA has been demonstrated to enhance in vivo translation and thus reduce the undesired immune activation. The main target for purification is the removal of double-stranded RNAs, which, if left within the vaccine solution, can cause several immunological complications for the patient. They are not effectively removed using traditional methods of purification, such as size exclusion chromatography or ion exchange chromatography. So we propose to use a newer method of purification developed by BioNTech RNA Pharmaceuticals, which can eliminate up to 90% of dsRNA impurity. This method utilizes cellulose powder with a buffer containing ethanol and permits the binding of dsRNA to the powdered solution. Advantages for this purification method are that it is fast, cheap, and scalable, in addition to having no toxic or hazardous waste generation. This purification method is different from traditional methods as it only requires standard laboratory procedures and does not involve any toxic chemicals or cell culture byproducts. Furthermore, relatively short purification time presents fewer opportunities to induce contaminants and microorganisms. Size exclusion chromatography can be used as the next step to filter out any remaining contaminants, specifically targeting any residual enzymes or transcript byproducts, such as uncapped mRNA. While purity data from mRNA vaccines is not readily available, we will base purity values based off recombinant DNA vaccines. For the recombinant DNA vaccine, the maximum residual amount of DNA should be below 10 nanograms per dose, with DNA sizes below 200 base pairs for parentally administered vaccines, according to the FDA. Furthermore, the WHO recommends protein purity of at least 95% for the malaria vaccine. Also, vaccine manufacturers do not release a lot of specific values, but Novartis recommends an endotoxin level below 20 EU per ml for recombinant subunit vaccines. Purity data from Merck reported protein purity of 98% and residual DNA content of 0.84 nanograms per ml in the HPV vaccine. Finally, an important aspect of mRNA vaccines is the delivery. Naked RNA is prone to nucleus degeneration, but is also unable to pass through the cellular membrane due to its negative charge. Currently, the most promising approach to solve this issue is the use of LNPs, which utilize phospholipids and aminolipids that condense nucleic acids at low pH values. In addition, PEG lipids provide steric ability, and cholesterols can be added for vesicle stability in vivo and in vials. Additionally, due to the issue of thermal instability, the mRNA vaccine will need to be lyophilized. Previous research has demonstrated that lyophilization mRNA vaccines retain its biological activity in a 25 to 20 or in a 5 to 25 degrees Celsius range for 3 years and for 6 months at 40 degrees Celsius. Lyophilized naked mRNA can remain stable for 10 months while refrigerated at 4 degrees Celsius. The global influenza vaccine market size was valued at 3.96 billion in 2018 and is projected to reach 6.2 billion by 2026. North America dominated market shares, which is good news for our US-based company. Because there are other technologies available and other vaccine platforms available for influenza, we don't expect this will be immediately replaced by other candidates, especially because it will take time to develop and research. Thus, the target market will start off a lot smaller than the total available market, but due to the large market size, we expect that there will be a stable revenue stream. In comparison to our competitors, Moderna, which has developed a competing mRNA vaccine, received nearly $1 billion from the Biomedical Advanced Research and Development Authority and has $1.5 billion contract for 100 million doses, bringing its price to around $50 per patient or $25 per dose. Pfizer and BioNTech have set the initial price at $19.50 a dose, which comes to $39 per patient. Assuming testing for continuous production and for purification is a success, we can file for a patent for the novel design, and once approved, license can be set up with other companies which may have higher production capabilities. This would help us keep up with an increasing demand, reach larger market segments with more advanced distribution capabilities. According to a 2016 study, the total development cost can range between $2 to $3 billion, with a 1.4 billion coming from out-of-pocket expenses and 1.2 billion associated with time expenses. The most costly is the preclinical studies and early research, which have ranging values from a couple of million to billions of dollars. Another thing to consider that even after approval, there are still additional R&D costs. Assuming that one dose takes $1 to manufacture, and if this vaccine is sold at $20 a dose, profit margins will be high. However, because this new technology will most likely be introduced gradually, and because development costs and timelines may vary among different companies and candidates, it is difficult to confidently predict when this technology will become revenue neutral. However, based on a simple prediction that all 500 million doses are successfully sold in a year, the profit would be 5 billion a year. This is unlikely, but demonstrates a huge potential in this field.